Have you ever really thought about how a bird is built? I mean, how a tiny, flat little speck of cells just sitting on top of an egg yolk can somehow transform itself into a complex three-dimensional animal. It is one of the most amazing engineering feats in all of nature, a process of, you could say, biological origami called gastrulation. So let's get right into it.
So yeah, this is the big question, right? It just seems impossible. You have this simple, two-dimensional sheet of life, and it has to organize itself, fold itself, and create every single part of a living, breathing bird, all while it's stuck on top of this massive food supply, the yolk. How on earth does it work?
Okay, here's our roadmap. We're going to tackle this in 5 steps. First, we'll look at the yolk problem, then how the 2-layer blueprint gets made, after that, building in 3 dimensions, followed by creating a self-contained world, and finally, we'll zoom out and see how it all fits into the idea of one goal, many designs.
All right, first things first, the biggest hurdle for this little bird embryo is, without a doubt, the yolk. The yolk is this huge, dense ball of nutrients, and it just completely gets in the way of the normal, standard methods of development. So check this out. In an animal that has very little yolk, like a sea urchin, the cells can just do a simple fold inward. It's a process called invagination, and that's how they start building layers. Easy enough. But for a bird embryo, that giant yolk makes a simple fold-in totally impossible. It's like trying to fold a piece of paper that's been glued to a bowling ball. It's just not going to happen. So you need a whole new strategy. And here is the bird's absolutely elegant solution. Instead of even trying to move the yolk, the embryo just works around it. All the action, all the construction happens in this super thin, flat cap of cells called the blastoderm, and it just sits right there on the surface of the yolk, leaving the yolk completely out of it for now.
Okay, so the embryo has its construction zone, that flat blastoderm. Now it's time to drop a blueprint. This next phase is all about organizing that flat disk into a couple of really fundamental layers.
And it starts off pretty simply. First, that blastoderm splits into 2 sheets. You get an upper layer called the epiblast and a lower one, the hypoblast. But then something incredible starts to happen. Cells on the surface of that top epiblast layer, they start to migrate, all moving toward the center. This creates, well, it's kind of like a cellular traffic jam, and it forms a visible thickening, a line, that we call the primitive streak. Now, this primitive streak isn't just some random line. You have to think of it as a highly organized gateway. It's the main construction entrance for cells to move from the surface down into the interior. In other animals, this entrance might be a simple little hole. But here, on top of this yolk, it's this incredibly specialized streak, custom built for the job. So as these epiblast cells dive down through this primitive streak, they start forming 2 brand new layers underneath. The first wave of cells to go down forms the endoderm. That's going to become the lining of the gut and the lungs. The next wave of cells files in and forms the mesoderm right in the middle. That's destined to become muscle, bone, blood, all that stuff. And what's left of that original epiblast on the surface, well, that becomes the ectoderm, the future skin and the entire nervous system. And just like that, boom, the 3 primary layers of the body are all set.
Okay, so we've got our flat, 3-layered blueprint. But, it's still just a sheet. The next huge challenge is to turn this sheet into an actual 3-dimensional body. And this is where that biological origami really kicks into high gear. To get from 2D to 3D, the embryo literally has to lift itself up and away from the yolk. And it pulls off this amazing feat with a really carefully choreographed sequence of body folds.
The whole thing starts right at the front, where the brain and the face are going to be. A wave of tissue called the head fold. It just starts to rise up, almost like a little cresting wave, and it lifts that entire front end clear of the yolk. And with that one move, you've got a recognizable front end.
And next, as you can probably guess, a very similar movement starts at the complete opposite end. The tail fold rises up, lifting the back region away from the yolk. This establishes that clear head-to-tail axis, It defines the basic layout of the whole animal. And for the final move, to create the main body, you get these lateral folds that start pinching in from both sides. They move towards each other underneath the embryo, and they meet right in the middle, basically zippering that flat sheet into a complete tube. This is the exact moment the 2D blueprint becomes a real 3D body. And the result of all this folding is, it's just incredible. What was once just a flat disc is now a 3-dimensional cylinder. It's the basic body plan of the animal, a tube within a tube. Totally distinct and separate, sitting right on top of the yolk that's going to keep feeding it.
So, the basic body is formed.
But the job's not done. Now the embryo needs to build its own private, self-contained world. It needs a life support system that will protect it and sustain it inside that hard eggshell.
And it does this by creating a few specialized membranes. You get the amnion, which forms this fluid-filled sac right around the embryo. It's like its own personal shock absorber, its own private pond to keep it from drying out. Then you've got the chorion, which is the outermost layer, kind of like a big bag that encloses everything. And finally, the allantois, which is basically a waste storage sac for collecting things like uric acid so the embryo doesn't poison itself.
But check this out. This next part is absolutely brilliant. That outer chorion and the allantois waste sac, they actually fuse together. And this new combined membrane, which is full of blood vessels, pushes right up against the inside of the eggshell. And since the shell is porous, this membrane becomes the embryo's lung. It lets oxygen come in and carbon dioxide go out. Just an amazing piece of engineering.
So, we've watched our bird embryo get built. Now let's just zoom out for a second and see how this very specific, very intricate process fits into a much bigger, universal biological principle. Remember how we started? The huge amount of yolk is what forces the strategy? A sea urchin can just fold itself inward. But a bird has to rely on this incredibly complex migration of cells moving across a surface. They're two totally different methods, but they're both driven by different evolutionary problems that needed solving.
And there's a great quote from our source material that just nails this idea. It says, "Although many different patterns of gastrulation occur in various organisms, the end result is the same."
And that's really what it all boils down to. Gastrulation, no matter how it happens, is the moment that form and structure are born. It's the master process that takes a simple blob of cells and rearranges them into an organized, layered blueprint ready to build a body.
And that leaves us with one final, just awe-inspiring question. We've seen what happens, the migration, the folding, the layering, but we haven't even touched on how. How do all of these millions of cells know exactly what to do? What's the invisible force, the molecular signal, the genetic GPS that guides this entire incredible biological ballet? Well, that's a story for another time.